Thank you, everyone, and thank you for the invitation to present today. Very interesting talks all morning so far. So I'm going to diverge a little bit from the past talks and talk about some new things we've been doing. And to start with, it's just a general sort of interest of mine about the yin and yang of science technologies. There's always this drive to answer new problems, and those result in the development and applications of new technologies that we all apply in our laboratories. Things that you can think of readily include DNA sequencing, PCR, all of the above. But there's also the development of new technologies that then we think about as scientists that we can adapt or co-op into our own laboratories to apply to the various problems we might have. And as I've always heard said, scientists look for solutions to problems, engineers look for problems to apply solutions. So there's always this yin and yang, if you will, between the two fields, which I find fascinating. And that synergism, I think, pulls everybody forward in science. So today I'm going to talk about the application of two platforms that we've been using in our work that have not been applied in the field of toxinology to any great extent. And that's second harmonic generation, two-photon confocal microscopy, and spatial omics, which includes proteomics, transcriptomics, and as you heard this morning, metabolomics. So I will give a brief presentation on each one of these and how we're trying to apply these to some of the problems that we're looking at. So as a reminder, this is the canonical slide of a basement membrane. It's comprised of a variety of proteins that interact to form this network. And at some level, they connect to the cells below it. And then above it, if you will, they connect to the stroma, the collagen fibers, and other extracellular matrix proteins. And as you've seen along the plasma membrane, this basement membrane here is quite predominant. And then at some level, eventually it connects to the collagenous stroma above, and then the cells below. And about, 15 years ago or so, we started to look at other ways to investigate the wounding and envenomation that we see. And we looked at proteomics in the exudates of those wounds. And this was first published as the wound exudate as a proteomic window to reveal different mechanisms of damage. We also have used this in human blister samples as well. And I think, as I'll show you a little bit later, they have provided great insight into what's actually happening when venom meets tissue in a victim. So this particular table is just a summary of what we've found in exudates from 3 different snakes. And if you look at this listing of all the proteins on this side, it's just a shopping list of what can be found primarily in the basement membranes, if you will, of tissues, as well as some stromal tissue, stromal proteins as well, such as collagen 1 and collagen 3. Now, keep in mind, all of these are fragments because they're soluble, they're found in the exudate, and predominantly that means they've been proteolyzed to some degree to give rise to that solubility and the fact that we can extract them and then look at them. So more recently, we've decided to go back and look at what happens in the case of muscle. As you know, in many mouse models of envenomation, you inject the venom into the muscle, and then either you look systemically for effects or you look at the local effects of that injection. And if you recall from your high school or college days, the structure of a muscle, you have a muscle here. It's surrounded by this epimyosin. It's a matrix of sort. And then you have the muscle fascicles here, within which you find the bundles, and they also are then surrounded by a perimyosin and an endomyosin sort of extracellular matrix. Now, when you look at the composition of those matrices, which is actually called the myomatrix, there's a wide variety of proteins that are involved in giving rise to structural organization to the cells below, if you here, and to the matrix above, the collagens, the fibrillar collagens, 1, 3, etc. Many different molecules play a role in that probably very precise organization, precise stoichiometry of these proteins. The ones I've highlighted in yellow are all that we found in various different exudate studies that we've done. So clearly the implication is that when you treat muscle, in this case with venom, or perhaps with individual snake venom metalloproteinases, you're very likely disrupting this to a very large degree. To what effect? Well, we know ultimately there's necrosis of the muscle, there's disorganization of the muscle, and ultimately, in many cases, regeneration of the muscle, depending on the severity of the wound, as well as the type of venom that's injected. So in thinking about that, in other words, the impact of venom and or toxins on collagen and muscle structure, as I think you all are aware, muscle damage caused by envenomation is a point of significant morbidity in some victims. Although the cleavage of the fibril collagen during envenomation is not immediate, it does occur later on in the sequela of the pathophysiology, as shown by ourselves and others, where you do see eventual collagen fragments being produced, but those are likely due to the endogenous MMPs either being synthesized or activated over time, because there's really very little, if more likely no, fibrillar collagenolitic activity in venoms. Well, as I said, and as I've shown, venom and snake venom metalloproteinases do degrade those ancillary proteins that have been shown to support collagen structure. And some of those proteins include collagen 6, collagen 7, collagen 12, collagen 14. The latter two are facet collagens, collagens that are known to be associated with fibrillar collagens, decorin, laminin, nitrogen, and many of the others. And the question that comes to my mind and our minds is, what actually happens to the fibrillar structure when those proteins are cleaved? Does the structure of the collagen, the fibrillar collagen, remain the same, or does it change by virtue of the fact that these ancillary proteins are degraded? There's a much literature that you can find regarding whether there's lack of some of these proteins or mutations of these proteins in human populations that give rise to a variety of muscular dystrophies. So you sort of wonder what could be the effect on the collagens when those are removed, because in the case of humans, they've been shown to be critical for the synthesis of that fibrillar collagen, but it's not exactly clear what role they play after the synthesis. And so what we postulate is that you may see a change of the collagen structure, the fibrillar collagen structure, that ultimately could give rise to issues associated with the healing and regeneration of that muscle tissue, and ultimately impact the morbidity of the muscle with regard to the victim. So to this, we turn to second harmonic generation. Basically, it's a multi-photon, in this case, 2-photon technique where you illuminate the sample with 2 photons. And by virtue of the structure of the molecules, in this case, fibrillar collagen, you get a fluorescence out of it about double the intensity of the original photons. And so this is from muscle. This is with the photons adjusted for collagen structure, giving rise to this green image. And what you can see at different Z stacks through the muscle, the structures of the collagen. Clearly, you would all recognize this as fibrillar collagen, sort of what you've seen in textbooks, as well as this. This is from a little bit higher Z stack, shows a fuzzier structure. And according to what I've read about the physics of secondary harmonics, that's because the intensity changes as the linearity of the molecules change. So sort of just shows you what, if you will, the control is. So you inject PBS in the muscle and then look at it from there. This is a case where when you use these particular settings for the lasers, you also get another color being fluoresced. In this case, it's colored as magenta, and it actually is from erythrocytes associated in the tissue, in the muscle, and erythrocytes themselves will show up. So this is all the way through the Z stack of the tissue, all together, showing both the green and if you will, of collagen and the red of muscle. You can see some sort of, if you will, maybe organization at a perpendicular angle of the collagen at this state, but still that spidery sort of collagen, particularly as you look in between the surface level and down below, you can see the spidery collagen. We think one is more representative of the perimyocin and the other of the endomyocin. Let's see. Yeah, I think, let's go on, I think. We're going to go on. Yeah, and so here we just show, as we've done, we've taken a venom, I believe this is Bothrops asper, and we've looked at the effects on muscle. The top is the control, where we have the composite image, simply the channel with the collagen and the muscle bundles. At 1 hour with the total venom, you can see a more of a striated pattern of the collagen, not as spidery, if you will, as it was. Here it is with just the collagen channel. In the case of the muscle, you can see the muscle is still more or less organized, but there's clearly erythrocytes showing up in the muscle, probably representative of hemorrhage in the environment. When you move out to 3 hours, it's much more pronounced in terms of the striation. It's more compacted. And over here, you see a very much starting to separate the muscle bundles, more hemorrhage, et cetera. If we look at a P1 snake venom metalloproteinase, if you look at just the collagen channel, somewhat similar if you kind of look carefully to what the control might be. But what you can see here is that there is disruption of the underlying muscle tissue and erythrocytes. However, when you go out to 3 hours, there's pronounced change in comparison to the control, where you see this condensation of the fibers into these strands, sort of kinky strands in this case, rather large, very much different from what is seen in the venom itself, and very disrupted muscle structure underneath. For a P3 snake venom metalloproteinase, you can see some level of striation appearing here. And you can see again disruption of the muscle. By the 3 hours, again, it's highly compacted, even more so than what you see here. But you also notice that there's something different. The muscle bundles are not nearly as obvious in the composite image. And over here, you can see they're totally disrupted. Now we can do a variety of clever microscopy imaging with this. And this is just going through the Z stack. We're starting on the outside and do a side shot. And then we go back. And then we're going to scale down through the Z stack, through the collagen. down into the muscles, and then we'll pause and come back up to the surface. And so come back up here in a minute. Suspense.
Just whilst we're waiting, you have 5 minutes left, Jay.
All right, thank you. Well, I don't know what happened here, but I do want to show you this one image here. Is if we make a cartoon out of this and we go in from the side view, what you can see is a separation between the two layers. So there's disruption of the muscle myomatrix. There's some herniation, if you will, of the muscles into the stroma above, and this disruption that is suggested. So our speculations is that normally there's a tight junction between the perimyosin and the muscle that's probably disrupted. The spidery orientation of the collagen fibrils is being disrupted. The P1 and the P3, I will argue to you, gives rise to a different structure of the fibrillar collagens over time. And that's probably because they're targeting different molecules. And I would say that this probably has an impact on muscle healing. I'm going to briefly tell you about our final area of interest, and this is looking at spatial analysis of the effect of venom in muscle. We take a region of interest, in this case, we're looking at Desmond, and we go in and look at the proteomics of a particular region. What we're looking at is the disrupted necrotic region, adjacent region, and then distal region. We've decided to focus on the immunology of this, primarily because of wound healing, and using various panels of probes, we can see that there are markers for M2 macrophages, particularly at the later times, which is what one might expect. We also see the intravasation of neutrophils over time, which one would expect to be involved in the clearance and removal of dead tissue and ultimately giving rise to angiogenesis. And again, in terms of tissue regeneration, we see cell proliferation taking place in the necrotic region. We see tissue regeneration by virtue of the presence of fibronectin, and also a marker for myoblast regeneration is upregulated at that 24 hours. So that's just a brief insight into what we're doing. We're carrying these out to longer times to try to better understand the impact on regeneration by different venoms and perhaps in different hosts. The muscle of the mouse is perhaps not the best model. So summary again, and all of that is that we're able to get a pretty good immunological picture of what's happening in the wound and how regeneration and healing is taking place. And we think it's going to give us insight into what venoms and what sort of tissues may be more amenable to morbidity or may give rise to morbidity and more amenable to various treatments. I want to thank the usual suspects at our shop in Virginia, Natalie, Anna, and Pat. And then in Costa Rica, Dr. Gutierrez, Dr. Rucavado, and Dr. Escalante. And then at Butantan, Dr. Ana Mora da Silva and Dr. Sir Jimenez. So with that, I hope I'm close to on time and I'll quit.
Thank you very much. Do we have any questions here in the audience? No? Anyone online who has a question? Could you unmute yourself? Okay, I'll ask a very quick question. Just out of interest, have you tried any SVMP inhibitors in your imaging? And do you think they might help alleviate the endogenous MMPs contributing to this pathology?
Yeah, no, we haven't. It's something, all of these things are opening up to us. This is actually fairly easy to do, believe it or not. And we're still trying to sort out actually the physics behind it. But all of these things we're really looking at. To me, what's more interesting is how these may be applied to our understanding of uh, human disease, the muscular dystrophies, and what proteins are really being affected by these toxins. Because as I said, I believe they have very different targets, and we could have a long discussion on that. But it gives rise to these different structures, some of which are probably more readily regenerated or healed or not, or maybe cleared by the MMPs. So as I said, I think there's some very interesting human correlates here that we're going to try to follow up, as well as the venom work.
Great, thank you.